All right, so we are now finally getting into the animals and able to talk about the different animal phyla out there. The big picture, there's a whole lot of animal phyla recognized by science. Probably one of the groups that we have the most knowledge about, not because there's more of them, but because they, for most of us, are more interesting than the other groups we've been discussing. So lots and lots of stuff's known about animals. What I'm going to do is keep it very broad and general. We have 26 different phylum recognized, and that number is changing. We do not have nearly the time nor the opportunity to explore all these phylum. So what I'm going to do is kind of highlight certain phylum. We'll talk about some representative examples of them, kind of what makes that phylum unique and special. In a couple examples, we'll go down into class, maybe even drop down to order occasionally, but by and large, we're keeping things at the broader phylum level. So again, this is just kind of a highlights reel. If you wanted to learn more about a particular phylum, you could take an entire semester over just one phylum or a handful of phylum of animals because we have that much knowledge. So think about this as your sampler platter. If you're interested in animals, which animals do you really like? What animals might you want to do further research or studying on and pursue it? Again, lots of opportunities to explore and discover new knowledge and new information. Okay, so our first phylum is going to be phylum Porifera. Those are the sponges. We're going to start with the simplest animals and work our way up the evolutionary chain, talking about different phylum as we go, and again, kind of highlighting some of the key members of the phylum and what are really kind of the key characteristics at the larger phylum level. And again, maybe we'll drop down into class a few times. So, all right, so an easy way to look at phylum, use the big picture, kind of the roadmap here with this table. Table's right out of the textbook, 33.1, lists out some of the key phyla. And again, don't worry about all of these. And I'm not doing them in this order. Why they placed them in this order, I could not tell you. But this does give us kind of the general overview. Phylum, kind of a typical example, key characteristics, and then approximate number of species. I'm not worried about you guys remembering those numbers, like how many species are in this phylum. It's a moving target. We're constantly discovering new things. And a big thing that's changing this is molecular data. Molecular systematics is reorganizing the animal phyla and how we place animals within the different classification levels. So don't bog down in the little fine details. That's not what this is all about. This is about big picture stuff. And again, as we eventually move to ecology, how do all these phyla tie together with each other? But then let's go back and tie them into the fungi, the protists, the plants, the bacteria, all those other groups we've discussed. Okay, so 33.1 and 33.2 list out kind of the key phyla. We're only going to highlight a certain handful of these things. The kind of the bigger picture roadmap here, this is the modern phylogeny of animals, is laying them out in this fashion. So our ancestral animal, let me get a little pen, arose back here, and then diverged, gave rise up this line to choanoflagellates. Those are the protista. We see the line breaking here, and then we see it diverging here and coming up into the sponges or porifera, and then some went this way, came up, split, came up here, gives us Cnidaria, Ctenophora, and so on. So this is kind of the approach I'm going to take when we start walking through the phyla, the evolutionary approach versus how they listed them out in table 30, the table we just saw in the previous two slides, 33.1 and 33.2. Okay, so here's our roadmap, big picture roadmap, kind of what we're doing, how we're doing it, and we're going to walk through these guys. I'll talk about key characteristics, things like that. Something to mention, we have these groupings up here, not classification levels, not a domain, kingdom, phylum, organizational levels that I will bring up. So we got Parazoa, we have Eumetazoa, and then remember, protostomes, deuterostomes. Hopefully those two look and sound familiar. And then within these groupings, we have other groupings, Sporilia, we have Platyzoa, Locotrophozoa, try saying that when you're not drinking, and then Ectozoa, and then we drop down to this level here. This is what I would be calling the phylum level. All right, so let us begin. First group to discuss, Porifera. Porifera go into Parazoa. Now, that's the general grouping or organization that we're going to put them in. Again, don't call that a kingdom or a domain or anything like that. Grouping, just like protostomes. Now, Parazoa, let me get a little text box here. Parazoa, actually, I'm going to go to the next slide. Parazoa, text box. All right. Parazoa means animals that lack true tissues. These are also going to be animals that lack defined symmetry. All right, so our sponges go here. At this point, sponges are the only ones that go here. So phylum Porifera are the only animals grouped under Parazoa at this point. Sponges don't have true tissues. They have cells, but what's really crazy and really cool about these guys is the cells do not become defined to just a single task. All right, so here's the example. Here's your sponge. Water goes in, and then it goes out. Anything in the water gets captured by these little cells inside the sponge. And I'll show you a better picture and talk more about this in the next slide. And then the water goes out. All right, so now you would think, okay, they're multicellular organisms and they eat. They're filter feeders. That's what we look at ecologically with sponges. And pretty much anything floating through the plankton load, they're going to consume, right? Just by default. So think about plankton. We talked about plankton. We've discussed what makes up plankton. So you have algae, and then you have the tiny little animals or other protists in the plankton load. So if you're a sponge, you're eating whatever's floating in that water. Could be producer, could be consumer. This qualifies you as an omnivore. Because anything that comes into your body, you can filter and consume as an energy source. So you're going to ecologically be a filter feeder. Specifically, you're an omnivore versus a carnivore, herbivore, etc. All right. So you have all these tissues or these cells that work together to do this job. And normally when cells work together to do a job, we say, oh, you have tissues. But what's crazy cool about sponges is take a screen. This is supposed to be a piece of wire screen, like a Windows screen. All right, so open your Windows up. You got a Windows screen there. Probably looks a whole lot better than this. But if you take that sponge and smash it through that Windows screen, you get all these little pieces on the other side. You basically smashed it up and pushed it into all these tiny little individual cells. Give that sponge enough time and the right environmental conditions, and it reforms. And it starts working again. Water goes in, filters food out, water comes out. Okay, so what's happening here is all the cells are getting mixed up, and then as they reform, they basically take over new jobs. They're like, oh, I used to do this, now I'll do this. Our bodies cannot do this. You can't take your arm, shove it through a blender or a wood chipper, and then have the cells reform themselves and take over new roles. They're specialized into specific tissues. Cell types form tissues in the other animals. Sponges, it doesn't work that way. So this is why they go into their own special grouping called parazoa, because they lack these true tissues. And if you remember symmetry, we talked about symmetry in the last chapter. You have radial, where I can cut you like a piece of pie or a pizza. And then you have bilateral. This is supposed to be our squirrel. And depending on which way I cut you, I get one view this way, a different view this way, etc. No, sponges don't work that way. There is no symmetry to a sponge. They're all different kinds of shapes and sizes and features and so on. All right, so again, this is where these guys go. But they are pretty cool. They are animals. There's about 7,000 recognized marine species of sponges living in the ocean. There's about 150-ish freshwater sponges. So believe it or not, Lake Springfield, you can find sponges. Let's go down to the ocean. Yeah, we're going to find sponges. Because they are filter feeders, they can live in deeper water because they don't need sunlight. They're not photosynthesizing. They're going to be down in deeper water. And I tell you, one of the crazy things I've seen while I've been diving, one of the biggest predators to sponges are turtles. Sea turtles. They love it. Here comes Mr. Turtle. Chomp, 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 chomp. That's supposed to be its mouth. So work with me, guys. You know my drawings are terrible. Trying to do this on a mouse, on a computer like this, it's even worse. Get a little turtle tail. So what happens here is the turtle comes along and it chomps up the sponge and it shreds it up into little pieces, and these pieces go drifting away. But as long as there's enough of the pieces there, or the piece is big enough, that sponge can actually regrow. So it's pretty cool. They can reproduce asexually. Let me clear out some of this. So they can do asexual reproduction just by fragmentation or being ripped apart by a predator or by wave action or by anything else. Whew, exhausting erasing all this. Anything that fragments their body up can allow them to do asexual reproduction. Oh, typo. Fragmentation. They can do sexual reproduction. Oh. So they will release, and I'm going to find you a really cool video over this. They release eggs and sperm into water column. And we call this a broadcast spawner. So it's this little cloud of eggs going up, or little cloud of sperm going up. One sponge doesn't produce both at the same time. They are hermaphrodites. They will produce both, but you don't send out your boys and girls at the same time. One today, a different one tomorrow. That way you cross-fertilize. You don't fertilize yourself. But broadcast spawners, releasing it up into the water column, and what we'll learn is most of the spawning is based on lunar cycles. The moon at a certain position causes the broadcast spawning. Okay, so I'm going to go out, find a little video, show you guys a video in the next lecture, talk about basic sponge features and characteristics, and give you a little bit more of an overview of how sponges work and Phylum Porifera.